Hi, my name is Shannon Murphy, and I'm a Ph.D. candidate in the DOOR lab at Cornell. I work with Vibrio cholerae, a gram-negative bacterium that causes the diarrheal disease cholera. Vibrio cholerae has horizontally acquired a few genomic islands, and these are thought to confer a fitness advantage either during life inside the human intestine or within its native marine environment. But we still know surprisingly little about the contents of these islands. But today I'd like to expose some of their secrets. One of these genomic islands, VSP2, is strongly associated with the dominant pandemic strain, but its 26 kilobase is a mystery. To this day, it's still unclear if or how it enhances fitness in either the host or in its native marine environment. Most of the genes encoded on VSP2 are not expressed under standard laboratory conditions, suggesting that their induction requires an unknown signal from either the host or the environment. One of those cues might be zinc limitation. Zinc is an essential micronutrient required as a cofactor in many metalloenzymes, but bacteria are often metal deficient inside the human host and in other natural reservoirs. In previous work, I found that this VSP2 gene, Shy-B, is a cell wall hydrolase that is only expressed when cells are zinc starved. This led me to wonder, could other components of this island be activated by low zinc? Here's how I found out that the answer is yes. While studying that cell wall hydrolase, I started looking more into how bacteria cope in low zinc environments. When zinc is plentiful, the transcriptional regulator called ZIR forms a complex with zinc and binds to promoter regions, repressing transcription of downstream genes. But under low zinc conditions, ZIR dissociates, allowing for expression of things like zinc importers. If we knock out ZIR, we get constitutive expression of all of those starvation genes. And when I was working with a ZIR mutant, I noticed something odd. This strain forms an aggregate when cultured in minimal medium. You can see in the video that the supernatant is very clear, and the cells have loosely accumulated at the bottom of the tube. My hypothesis was that some member of the ZIR regulon is mediating this behavior. But when I checked every known ZIR target in cholera, none of them were required. So I devised a screen. I took the ZIR knockout and mutagenized it with transposons, and repeatedly subcultured this library until the pellet stopped forming, and mapped the site of transposon insertion. What we found is that the pellet did not form when motility genes are disrupted. These included hits for flagellar components, as well as chemotactic signaling. This suggests that this aggregation is an active, motility-driven process. After sequencing many candidates, we still hadn't found anything that might be ZIR-regulated. So we devised a second screen, where we pre-filtered the library to select for mutants that could still swim. Through this workflow, we identified required genes that were overshadowed in our first iteration. And to my excitement, both of them were encoded on VSP2. These included a chemoreceptor, which can sense a chemical ligand, as well as a transcriptional activator belonging to the Araceae family. Are these VSP2 genes ZIR-regulated? If we compare transcript abundance, we see little to no expression in a wild-type strain, but significant induction in a ZIR mutant. It just so happens that this operon with Araceae, shown in green, is an obvious candidate for ZIR regulation. And we can tell by just examining the sequence of the promoter, which contains a highly conserved ZIR binding site. The alignment of that promoter and the sequence logo is shown here. But the chemoreceptor in blue does not have one of these ZIR binding sites. Could it instead be regulated by Araceae? To check, I built a Laxi transcriptional reporter for the chemoreceptor and measured its response to Araceae overexpression. And here you can see that Araceae causes a significant induction of this gene. In summary, this aggregated phenotype, while strange, has helped us to expand the known ZIR regulon in cholera to include a transcriptional activator and 2 other open reading frames. It's also helped unearth some of the secrets of the Vibrio 7th pandemic island. Through transcriptomics, we now know that a total of 9 genes on the island are expressed during zinc starvation. And we are also learning more about the island's functional capabilities. We know that this chemoreceptor leads to aggregation in nutrient-poor medium, and in future work, I'll be investigating the ligand or signal that this chemoreceptor responds to. Finally, my goal for this project is to put this phenotype in the larger context of cholera biology. Cholera undergoes many lifestyle transitions between motility, biofilm formation, and then dispersion. One hypothesis I'm exploring is that the zinc-regulated VSP2 genes might help assist in these transitions. If you'd like to chat further about this work, you can connect with me via email or Twitter. Thanks.